Okay, so today I will make a video to show an overview on the first Galaxy steps from the moment that we upload the data and then we create a collection of the data. We initialize a workflow. This means the steps that we need to perform an analysis. Today we will not do all the steps, but we will just show the principle how to create a workflow. And when we create this workflow, we use some tools. Therefore, I will show how we set the parameters of these tools. Even though I will show the steps just for a couple of tools, or even only one tool, a couple of tools, let's say. So I will demonstrate how to make a workflow using the tools like Falco, FastB, and Trimomatic. The same idea applies for other tools that we will see in next classes like BWA, MIM, and some tools of the suite SumTools, and so on. Okay, let's start. Now, currently in the Galaxy we have already done some things, so I have a full history here. Therefore, to demonstrate these steps, I will just make a new history. And actually, it's advisable to create a new history for every new project that you are doing. Don't have everything on the same history. To create a new history is very simple. You just go here on this plus sign and you click and you make a new history. You can change the name of the history here, just pressing this pencil. So the history, we will call it yeast demo. You can write whatever you want. You can write some description here, whatever you prefer. Okay, now we have the history yeast demo and it's empty, still empty. So we will go to do the steps. As we see here, the first step is to upload data. To upload data, usually we go to some database. In the class, we use the database ENA, European Nucleotide Archive. It's very easy to find it in Google. You just go to Google and you write European Nucleotide Archive. And it's the first link usually. It's a database supported by the EMBL, EBI, and it looks like that in the first page. Okay. So we are here, I close this tab. And here we have two search areas. In the first one we can write some text, but if we know the accession number of the study that we want to get data from, we can write the accession number here. Okay, let's go to search now. So in the class we use some data that are related to flour and wine making. It's yeast. So if we go here to project, sorry, it should be this, I don't see it here. Let's write it's this one, the Geno Wine project. Okay, so when we do that, we see the data here. It's actually 17 datasets. 10 of them are shown in the first page, and if I click here, I will go to the second page and I will see 7 more. Now, here we can choose the columns that we want to be visible. So if we go here on the right, for example, we see some additional information. The information that we want is actually here on these links. These are equivalent links. We will need actually the links from this column, the FASTQ files from an FTP server. Now, there are 17 datasets, and it's per den datasets, which means that there are 34 files. And it will be a bit cumbersome to download all of them one by one. What we will do is the following. We go here to TSV, and we download this whole table as a tab separated values file. I will just put it here in downloads, temporary directory. I open it, and I open it with the TXT viewer. My viewer is called text editor. In Windows it could be the notepad, the text pad, whatever you have. It's just a simple TXT file. Let's see where it is. It's not here. Let's open it. Check it. Okay, we open now the file with a text pad. It's called actually gedit in Linux. So this is the same information that we had in this table here. For example, you see here that the name of the study is, the accession of the study is PRJEB6529, and this information should be present somewhere here in different column though. It's this one. This is the name of the run, this is the name of the sample, this is the name of the experiment, an ID of the study, the species name, the technology used for the sequencing. It's Illumina HiSec 2000. It's good to remember this because then we will need this information for trimming. And then some files here, and here we have some links, URL links, and we see actually that the links for the forward read and for the reverse read, they are joined together and they are separated by a semicolon. Now what we will do is that we will open this in an Excel file. I will use Google Sheets for that, and then I will separate this URL into two URLs, this one and this one, and then I will make them be proper URLs by adding FTP : / in the front of each link, because here this is omitted as we see. Okay, so I select all, Ctrl A, I copy it, I go to Google here and I paste it. Now this is a more proper view than what we saw in the text edit, and we need this column. I just copy it. So here it's the whole information, just a column. The column is too narrow to see everything. And you see here is the whole information here. I just copy it. They go somewhere here. Not important where exactly I will go. And I paste it. And we see actually that the two links are here and they're separated by this column. And what I do is that I choose all of them, and then I go to tools or to data, maybe data, and then split text to columns. So I'm going to split this text into 2 columns, and the separator will be the semicolon. It's semicolon because here I see a semicolon, and here there is a semicolon, and so on. Okay, now I split in 2 parts. I get this part and I put it under the first one. It's cut and paste. Here I have everything, and then I choose again these cells and I replace with Ctrl H the FTP with FTP : / FTP. The idea here is to put this part of the URL in the front of this text. Okay, please be sure to do that only once, because if I will redo it, then I will replace again these FTPs with FTP : / and of course then the URL will be wrong. Okay, now I can get all these URLs, copy, and I go to Galaxy to upload the data that correspond to these URLs. To do this, I go here to upload. This window pops up, and then I go to paste fetch. I leave this new file as it is, and then I paste the URLs here. I have a quick look to see that everything is fine. Sometimes some strange things might be added here at the end. Everything is good. Everything looks fine. Okay, I don't change the file, the name here. I just leave it as new file. And the reason is that if it is like this, then the names of the files here will be created by the name of the URL here. So for example, you will have a file called ERR560543_1FASTQGZ, which is what I want. Okay, in order not to load my my space here with too much data, and not to make it too long, I will just upload a few of them. I will not upload all of them, I will just upload one file, let's say. So I delete everything, I go here again, just take a pair of files. I will take let's say this 34_1, go here, I put it here, and 34_1, and this is the _2. Go here and I put it here. I remind that this is just a demonstration, that's why I just upload 2 files. Okay, and then I just press start and close. Now what's happening is that I gave a command to Galaxy to upload these files from ENA, from this URL that I had pasted there. Quite often the server, the Galaxy server, is busy, and therefore it puts my job on a queue. That's why you see that this clock there, it was telling me that I should wait for the job to start. Now it started, but of course it's not complete yet, and you see this thing rotating here, which means that okay, it's working, but it's not complete yet. When it will be complete, this will be green. If something will go wrong, this will be red, and you will see a triangle here. Okay, let's wait for a while until this will complete. Okay, so I see here that I had a mistake, an error. It didn't download it. I didn't manage to download this file, the _1 file. The reason is not clear. I see here the URL that failed. I can click on this info button and I come here, and then I can see in this part the URL, which seems actually pretty valid. In any case, I copy it, I go to upload data, I press reset because maybe you will see here the old URLs, and then paste fetch data again, and then I download this dataset again. Let's see what will happen. Okay, now I just re-upload this dataset and it's there. Okay, why is that 2 again? That's strange. That's very weird. Let's see what we did here. Eventually I put the correct URL. By mistake I had put the one with the 2 here. Now I put it with 1, and let's wait to see if we'll get the right file. Okay, now we have 1 and 2. I delete this thing here. And now we are ready to make a collection out of these 2 files. If I would have uploaded all of them, I would have 34 files, and this would actually be 17 pairs. Now I have 1 pair and 2 files. Okay, what I do is that I click on this tick button, I choose all of them, both of them, and then with all 2 selected, I go and I create a list of dataset pairs. Sometimes this option will not be visible for whatever reason. This is a Galaxy issue, but what you could do then is to make a reload. If you make a reload, the page will be reloaded, and then you will have to click them again, and then probably you will see this option here. So build list of dataset pairs. It recognizes automatically that you have a pair that corresponds to this file. You give a name here, and then create a list of pairs. So this is a list. It's a list of 1 pair in our case. And now it's time to go to the workflows. So what we have done until now is that we have uploaded the data, we have created a collection. And then it's step number 3, initialize a workflow and set up the steps for an analysis. Okay, workflows. We click here to create a new one. We give a name. Just analysis, and we are at this step. A workflow usually includes some inputs, the input files. In our case, the input file is a dataset collection, so we click here. You can imagine that actually this is this box here represents the input. And when we click on this box, a menu will open on the right hand side. This menu here includes options related to this box. So for example, it asks, is there a label, a name of this? Okay, yes, there is a name. It's actually the yeast file. Okay, I can just put some annotation here, and then since this was a dataset collection, it asks me what kind of collection it is. Is it a list, so many files together? It's actually a list of dataset pairs. So here we should change the list value to list of pairs. Okay, now it's fine. We close this. So this is the input. And then we go to tools here to choose the first tool. And the first tool will be the quality control on these files. So we'll do that with the Falco tool. It's a nice tool that actually it's a bit more modern than FastQC. And then again we click on this button, on this box, and this menu pops up here. We can put a label, but we see actually here that there is already a proper label called Falco. So we will not put anything here because this is fine. So Falco has several options. It asks if we have a list of contaminants, a list of adapters. We don't need this actually here because anyway we'll trim later on with FastP that will automatically recognize the adapters. It asks several things that we don't need actually to modify them. So the only thing that we will do is that we will say, okay, I will connect the output of this data set with the input at Falco, that is the raw read data from current history. I was looking here to see if Falco accepts a dataset collection, but it seems that anyway it will manage to fetch the datasets from the collection and will treat them as raw reads from the current history, which is fine for us because anyway the quality control is done separately for each file in the collection. And then the report will be an HTML file and a text file with information from the quality control. If we click on something, this will be the primary output. We can click many of them, but in any case, if we will not click on any of them, the output will be created anyhow. Okay, let's click them. The other tool that we will use is the FastP, which will perform the trimming and the removal of the adapters. So I will put it here. And again, I go here on the right hand side. I have clicked on the FastP, so this is the menu for the FastP. Tool parameters. It's not a single end, it's a paired collection. We don't want to merge forward and reverse reads. Let's go to adapter trimming option. Disable adapter trimming. No, we don't want to disable it. It writes here that adapter trimming is enabled by default, so it's fine. It will automatically recognize the adapters, at least for the main platforms, and since we have an Illumina HiSec 2000, it's pretty easy for FastP to recognize the adapters. So we leave these adapter trimming options as they are. Global trimming options. We don't want to trim a specific number of base pairs, either for input 1 or for input 2. So if we set up here, for example, 3, it will trim three bases from the front of input 1. If I put here number 5, it will trim 5 bases from the tail of input 1 and so on. But we don't want to trim a specific number of bases. What we want to do is to trim bases that have low quality. This is what we will do later. Okay. Then we have some filter options here. Disable quality filtering? No. Quality filtering is enabled by default. If this option is specified, quality filtering is disabled. So quality filtering does not actually do trimming. Quality filtering is doing the following. It marks each base as a good or bad quality. To specify the good or bad quality, we put a number here, which is the quality threshold. So the default is 15. If you have a value that is lower than 15 in the quality of the base pair call, then this base pair will be marked as a bad quality base pair. If you have several of them, then several of them will be marked as bad. Now, if the percentage of these bad ones is higher than this threshold here, for example, if I will put a 40 here, means 40%, then the whole read will be marked as bad quality read and will be removed. The same we can do with n, with calls as n. N means actually it's a very bad quality base pair call, and the machine could not decide which one of the 4 nucleotides it is, so it puts an n there. So we can leave the default values as they are, and then we go to length filtering option. So if we remove the adapters and perhaps do some trimming at the end, maybe the length of the read will be very short. And here it allows us to specify a filtering. So again, length filtering is enabled by default, but we can modify the length. So the default value here for the length required is 15. It's a bit low, so we can put let's say 40 here. Now, for some reason, it has also a maximum length cutoff, but we can leave it empty because default is 0 and means no limitation. Okay, we leave the rest as they are, and then we go to read modification options. So these are options that might modify the read. And actually, this is the trimming of the bad quality segments of the read, either at the 3 or the 5' end of the read. And we can go here, enable per read cutting by quality in front 5', default is disabled, so we enable this, and we can enable the 3' as well. 5' cut and 3' cut. Cutting window size, let's put 4, so 4 bases, and the mean quality will be around 20 or so. So now if a window of 4 bases has an average quality of 20 or lower, then this window, and probably whatever it is from this window to the end of the read, will be removed. Okay, so we leave it like that, and we close the menu. And since this is a collection, it accepts a paired collection, and this is a paired collection, we can connect these two things. Now this means that the input will be given both to Falco, which will be just a report of the qualities of the reads, and to FastP, which will be actually the first step for our analysis. Now, in parallel to the FastP, we can do, just for the demonstration purposes, we don't do it in reality, we can also use another trimming tool, which actually has been used for years in bioinformatics. And this is called Trimomatic. So FastP is a bit more modern, but maybe some students want to apply Trimomatic, so I will just show Trimomatic as well, even though we don't need to apply both FastP and Trimomatic. So here is Trimomatic. We don't change the label again because the name Trimomatic is just fine. We have paired end collection, and this perform initial Illumina clip step. It means that if I will find Illumina adapters, should I remove them? Yes. And we'll ask here what kind of adapters do you have. You want to provide the adapters by your own or to use standard? Use standard. Now remember that we had Illumina HiSec and we have perdent, so either we'll use this TrueSec3 here because it matches with perdented HiSec, or this one TrueSec with additional adapter sequences. Again, it's perdented, MySec and HiSec. Let's use this one. We leave the rest as they are. Actually, we leave everything as it is, and we go here to Trimomatic operation and we say, okay, insert one more. So the basic operation that Trimomatic is doing is use a sliding window of 4 base pairs and require an average quality of these 4 base pairs to be 20. If it's not 20, if it's lower, then it will remove everything from this window towards the 3' end. This is fine, and we want to insert one more trimming operation, one more filtering operation, and this is the min length. So we search here for the min len operation, and we say, okay, if something is below, let's say 40 base pairs, the same that we did in FastP, remove the whole rhythm. So everything else is just fine. Okay, this is fine again, and we can also connect this input here. Again, I remind you that we will not need in a pipeline, in an actual pipeline, we will not need both Trimomatic and FastP. We can have just FastP and then proceed to the next step with the output of FastP. The next step will be the mapping, by the way. Okay, and since we have the pipeline, we save it. So we click here. And then we go here and we click on the run. So when we click on the run, it asks for the input. Which file should be the input? Okay, the input, we can choose it here, it will be the yeast. It gives only this option here because this is the only paired collection that we have. And then we just click on the run workflow button and it started. It shows here the workflow that we have and the step at which it is currently. So this is a workflow pipeline. It's the best way actually to analyze data in Galaxy, and I recommend everyone to use them for the analysis. If something will be wrong, you will see here again something to be red and have a red triangle here.